and welcome to Intro 101. My name is Chris, and I am your neuroscience and psychology graduate student assistant. I'm here to assist you with the related science basics that you interact with on a daily basis. My goal for doing this is to help you start or continue to build a foundation in human biology and physiology that can better help you engage with the information you consume from your favorite health and wellness educators and influencers. Much like the way a graduate assistant helps prepare intro students so they can better engage with lectures delivered by their professors. Or maybe you're here just because you'd like a better idea of what's happening inside your birthday suit. Either way, grab a snack and a cup of tea or beverage of choice and stick with me for the next bazillion minutes because today we are covering a lot. Today's episode is Intro to Cell Physiology 101. We'll take a look at the tiny but extraordinary universe of human cells. I'll go over basic cell structure, anatomy, and function. I tried to fit nervous system cells into this episode as well, but it just got way too long and confusing, and that subject really deserves an episode of its own. But learning cell physiology basics will go a long way in setting up that scaffolding needed to understand how nearly every cell in our body operates, including nervous system cells. So today, basic cell physiology, and next episode we'll tackle the physiology of nerve cells. Future episodes will also look at the physiology of cells from other body systems as they work in conjunction with the nervous system. So hopefully today's episode sets you up with a solid understanding of how all of those cells function at a fundamental level. Before we start, I'd like to respectfully acknowledge with much gratitude that Intro 101 is recorded on the unceded and ancestral territories of the Kwantlen, Katzie, Matsqui, and Semiahmoo First Nations. Additionally, we here at Intro 101 believe wholeheartedly that science is for everybody. In fact, the richness, complexity, and strength of scientific research is astronomically improved by the inclusion of many varied voices. In an effort to help everybody have access to education, research resources, and representation across scientific fields the world over, I will be sharing a few organizations in the show notes that are working hard to promote and grant access to STEM education around the world. Please check them out, and if you are able, please support them in any way that you can. You can donate and share their work on your social media networks, or maybe volunteer your time. If you already generate opportunities that provide STEM access, education, and resources within your own communities, or support other STEM education organizations not listed here, please share them in the comments below if you'd like others to contribute as they are able. All right, welcome to class. Let's crack on with Cell Physiology 101. So, what are cells, and why do we need to understand what they do? Well, first of all, there are two main classifications of cells for life on Earth. There's eukaryotic cells and prokaryotic cells. Multicellular organisms like humans have eukaryotic cells, and unicellular organisms like bacteria have prokaryotic cells. There are some unicellular organisms that are also eukaryotic, and yeast is probably the most familiar example of unicellular eukaryotes. The main difference between eukaryotic and prokaryotic cells is that eukaryotes have membrane-bound organelles inside the cell, plus a nucleus. Prokaryotes do not. This will become clearer as we get into cell anatomy. Okay, so something wildly fascinating about cell structure and function is that for the most part, cells are similar across all living eukaryotic organisms. Cells are the smallest organized living units, and they are roughly the same size in humans as they are in fruit flies. In fact, humans and fruit flies share many of the same types of cells. And in the next series of Intro 101, we'll get into the world of scientific research, and we'll look at how the fruit fly is helping us better understand the underpinnings of many relevant human neurobiological responses, including alcohol addiction. So, same size cells in a fruit fly as a human, but the larger the organism, the more cells it has. Now, neurons, which are cells in the nervous system, can vary in size and shape within and between eukaryotic species because of the length of their axons or nerve fibers. I think the giant squid holds the record for the longest axon in a eukaryotic organism. If you know of a longer axon, let us know in the comments below. But the size of most other cells is comparable across all living organisms. So, what do these cells, these smallest living units, do? Well, firstly, if you remember from the first episode, cells provide structure to your body. Clumps of cells together form tissue, and two or more types of tissue form organs, like your brain, your skin, your muscles, your bones, and your eyeballs. The shape and structure of the tissue is due to the type of cell and the way it is gathered together to form the tissue. Sometimes that's clusters of cells, sometimes that's layers of cells. Secondly, cells carry out life-sustaining processes, but just like structure, their exact function is largely dependent on the type of cells that they are and what body system they are in. For instance, your nerve cells specialize in communication, your muscle cells specialize in contraction, your red blood cells specialize in carrying oxygen to your body, your intestinal cells specialize in releasing molecules that break down your food, but also have cells that specialize in absorption of nutrients. So again, cell clumps make tissue, and then two or more of those tissues go on to form your organs. And organs are recruited by each other to work together to form interdependent body systems that keep you alive and functioning. So those cells not only have to work within their own tissue, they also have to work in conjunction with other cells in other tissues so that body systems can operate as they should to maintain homeostasis. If you remember from the first episode, homeostasis is your body recruiting one or more of its systems to maintain balance. For more on homeostasis, have a listen to the first episode in the series. The human body is made up of roughly 75 trillion cells, and so far about 200 different types have been identified and categorized. There are hundreds of trillions of other cells in the human body, but those are guests called microbes, and that's a topic for another day. But the cells that make up your body systems all have specific functions. So some examples of body system cell types are, of course, nervous system cells like neurons and glia. Skin is made up of layers of epithelial cells, including keratinocytes and melanocytes. You have muscle cells, including myocytes. You have blood cells. One type is called lymphocytes. Bone cells include osteocytes. Fat cells are called adipocytes or adipocytes or lipocytes. Liver cells are mostly made up of hepatocytes. I don't know if I'm pronouncing any of these correctly, but if you haven't spotted it already, if it ends in cytes, it's referring to a type of cell. One of the main functions of your cells is to reproduce itself. It's probably a no-brainer, but you exist today because one of 280 million sperm cells found an egg cell, fused with it, dropped its genetic wad into it, which combined with the egg's genetic bits, and the two cells became one. Then the combined genetic material replicated itself so it could undergo a process called mitosis, in which the cell divided and created more cells, and they divided and made more cells, and so on. And those cell babies are called stem cells, and stem cells go on to become their specialized self in a process called differentiation, where the genetic material in its nucleus provides a kind of blueprint as to what it should be and do. And guided by this blueprint, it finds its way to other cells that do the same thing, and they kind of attach together by way of special proteins in the cell membrane. More on that in a minute. And that's how they become tissues and organs and whole body systems until you become your very you self, and this process continues to happen throughout your lifetime in most parts of your body. So, all that to say, cell reproduction is one function of your cells, and importantly, according to cell theory, a cell can only create another cell from an existing cell. Which, if you think about that for too long, like I do, you'll find yourself falling down thought holes trying to figure out how multicellular organisms began in the first place. Like, what was that first cell and where did it come from? How did it materialize if cells can only be born from existing cells? Lots of speculation out there, but there have been many lab studies that have attempted to sort out the process of initial cell formation. And if that interests you, I'll post a link in the show notes below and you can go from there. But curiously, what are your thoughts? Post them in the comment section below. Now, each of those cells that formed you have many of the same basic internal functions, also called intracellular functions. And then, as I'll keep saying, On top of that, they have specific functions that make it the type of cell that it is. But fundamentally, each human cell must be able to take in nutrients and turn them into energy, store that energy, make proteins and lipids and transport them, and dispose or recycle waste. Each cell must also contain genetic material, and again, that material informs the cell what kind of cell it's going to be and what it's going to do. One caveat here. Your mature red blood cells, also called erythrocytes, do not have genetic material, or most of the other intracellular bits you'll find in the rest of your cells, so they can't divide and reproduce. But your white blood cells, also called leukocytes, do contain genetic material and do also have the same basic intracellular bits as other cells. Lastly, the cell's most dramatic function is to know when to shut down operations and self-destruct. But even during that process, you'll find that it's very economical about its energy and waste footprint. But more on that in a minute. So, why should you understand how your cells function? Well, your cells are you. They are the reason you are alive right now. When they all cease to function, you cease to function. When some of them cease to function properly or prematurely die, this can lead to disease states. Cell dysfunction and premature death is at the root of many neurological diseases and disorders. Cell overproduction and unruly spread is at the root of most cancers. When immune system cells wig out and start attacking healthy cells, it can lead to autoimmune diseases. For less serious health-related reasons, you may have fitness or nutrition goals, or maybe you're going for massive muscle gains. Learning about your cells will help you understand what you need to do to enable your cells to support these goals in healthy ways. We have a symbiotic relationship with our cells. Decisions we make affect their operation. The irony is that it takes a bunch of cells to make those decisions, but there is a limit to what your cells can do. If we push them beyond their limit without supporting them, we pay a price. Even if we engage them as expected, but do not take care of them and make sure that they have what they need to do their regular business, we also pay a price. That's maybe a bit abstract, but if we put this in context of the many protocols available for you to achieve your health goals, understanding what cells need to function and how they function will give you some perspective as to whether those protocols will meet those needs or put unnecessary demands on certain cells within their systems while trying to improve others. When we understand how our cells function and what they need to function so our tissues, organs, and body systems can operate optimally, or even just regularly, we can learn how to take care of our bodies in ways that impact us at the cellular level. The more we understand how our cells function, we may even be able to look critically at a protocol and decipher how it should play out mechanistically at a cellular level in our own bodies. Now, that may be a tall order from introductory level instruction, but this will at least get you started. It will set you up with a foundation from which you can build your understanding. Okay, so let's get into basic cell structure and physiology. So every type of cell in your body is going to have mostly this basic structure, except of course red blood cells, as I mentioned earlier. So first, the human cell's tiny universe is packed inside a fatty outer layer called the plasma membrane. The plasma membrane is incredibly important to homeostasis. It has a big role in providing structure to the cell and protecting it, as well as allowing things into and out of the cell. It is also an important player in the initiation of activity in the cell that launches or maintains life-sustaining processes that keep the body in homeostasis. So how does the plasma membrane do these things? Well, first, the plasma membrane is mostly composed of two layers of fat molecules called lipids. And that's important to know because that helps maintain its structure, but also determines what can easily cross that barrier into the cell. The lipid bilayer is quite fluid, like oil, and keeps the cell dynamic and kind of shape-shifting. An example of this is the flexibility of blood cells that need to adjust their shape to travel from larger to narrower blood vessels. The actual lipid molecules in the bilayer are called phospholipids, and there are about 1 million of them in the plasma membrane of one human cell. Each phospholipid has a head and two tails, and the heads are hydrophilic, which means they love water. The heads are also polar, which means they are also electrically charged. Water is also polar, and the extracellular and intracellular fluid is mostly water, so phospholipid heads like to line up and make friends with these spaces, and that's kind of how the lipid bilayer is formed. The tails are hydrophobic and electrically neutral and want nothing to do with water, and by extension, the intra and extracellular fluid. So they hang out with each other in the middle of the lipid bilayer. The membrane is also packed with proteins, and that's also important to know because what materials can't easily cross the phospholipid bilayer must then seek out certain proteins within or attached to the membrane, and these proteins allow material into or out of the cell. There are also proteins on the surface of the membrane that act as markers that help the cell recognize what type of cell it is, which is crucial for the formation of tissue during development, but also plays a role later in the immune response's ability to recognize itself. And that's important in the world of autoimmune disease. So proteins called cell adhesion molecules, or CAMs, act like little interlocking hooks on the exterior of the cell membrane, and one of their functions is to connect cells together to form tissue, kind of like a zipper. The plasma membrane also contains cholesterol, and that helps keep the lipid layers stable and prevents the membrane from becoming too rigid and inflexible. Which, we could all use a friend like cholesterol. Okay, so quick recap to keep you tracking. The plasma membrane is made up of a phospholipid bilayer, the heads of which are electrically charged. It contains many different kinds of proteins that have many functions, including allowing passage for material to travel into and out of the cell. It also contains a bit of cholesterol, and that keeps it chill and flexible. So, what exactly wants to get across the plasma membrane into and out of the cell? Well, mostly molecules and ions. So molecules that are lipophilic are fat compatible and can freely cross the plasma membrane into and out of the cell. An example of a lipophilic molecule is a steroid hormone, like estrogen or testosterone. Molecules that are lipophobic are fat resistant, and they cannot cross the plasma membrane without transport across the membrane by way of certain proteins. An example of a lipophobic molecule is insulin. Now, ions have different requirements to cross the membrane. It has less to do with lipid composition itself and more to do with the electrical charge at the different parts of the lipid bilayer of the membrane. Also, ions need special channels made of proteins to cross the membrane, but more on that in a minute. So how do proteins in the plasma membrane know which molecules or ions to let in? Well, again, it depends on what the molecule is and what the cell's function is. Membrane proteins are very selective of what can enter the cell, and again, it's highly dependent on what the cell's specialized function is. Some proteins inside the membrane are carrier proteins. They transfer molecules into the cell by being open on the extracellular side of the membrane for a molecule to bind to its binding site inside of it. That binding site has specific features that align with a specific molecule and will determine what kinds of molecules can bind to it. For instance, glucose molecules can't bind to and be carried across the membrane by an amino acid carrier protein. It has to be a glucose carrier protein. Once the molecule has bound to the binding site, the carrier protein will change its shape by closing its extracellular doors and opening itself up on the intracellular side. It then releases the molecule from the binding site so it can enter the cell. Another protein in the membrane allows for pod-like structures inside the cell called vesicle transporters to secrete their business into the extracellular environment. This will come up again in a minute when we discuss certain organelles, as well as in the next episode when we'll discuss neuron physiology and neurotransmitters. Some proteins in the membrane, as I've mentioned, are ion channels. These will also come up again when we get into neuron physiology because they are an important part of neurotransmission, which is a type of cell-to-cell communication. But some key points about ion channels is that they only allow ions to pass through, and each ion channel is ion-specific. So only sodium ions can pass through sodium ion channels. And some ion channels allow their ions to just pass through into and out of the cell, and some channels have a more complex process that will become clearer when we get into neurotransmission. I know I keep saying this, but it's warranted. The number and variety of ion channels is dependent on the cell's specific function. The last membrane protein I'll mention today comes up a lot in health and wellness discussions, so you've likely heard of them before, and those are protein receptors. They have important roles because they bind with chemical messenger molecules called ligands and signal to the cell that it's time to initiate some specific activity. Again, the types of protein receptors in the cell membranes are highly dependent on what that cell's job is. But examples of ligands that bind with receptors in the cell membrane, and ones which you've likely heard tossed around by health and wellness influencers, are peptide hormones like ghrelin and prolactin, and insulin is another one I've mentioned already and will certainly be familiar to you. It's important to note that once the peptide hormones, let's say insulin for example, makes contact with its particular protein receptor, it still doesn't just get a free pass into the cell. The protein receptor has to kick off another set of events inside the cell to let the cell know this chemical messenger is coming in. It's called a second messenger pathway, and it unleashes an incredibly intense biochemical cascade of events inside the cell, which eventually leads to the fusion of the insulin molecule with the plasma membrane. We'll get into the second messenger plan of action in more detail in a later episode. Steroid hormones, on the other hand, are phospholipid friendly, and they can cross the membrane without assistance, but they do bind to protein receptors inside the cells. As we get to the different body systems the nervous system interacts with, we'll get deeper into the ways those cells behave. So, quick recap, the cell membrane is both an important barrier and a gatekeeper, regulating the movement of material into and out of the cell, and that regulation is sometimes mediated by proteins in the membrane if what's trying to get into and out of the cell can't just easily pass through the phospholipid bilayers. So already that's a lot. And that's just one component of the cell universe, and it has a dizzying amount of complexity to its function, and I've only given you a moderate amount of detail. So take a breath, hydrate, and let's keep going. Okay, so let's take a look inside the cell. What is this little universe the plasma membrane needs to protect? So inside human eukaryotic cells, there is cytoplasm and the nucleus. The cytoplasm is everything inside the cell that isn't the nucleus. I'm not sure why the nucleus is considered separate from the cytoplasm. I'm told it's because it's surrounded by a membrane, but there are membranous bits that are considered part of the cytoplasm. So if someone has a better explanation, please let us know in the comments because it's a little confusing. So the cytoplasm contains organelles and the cytoskeleton, and those are suspended in a gelatinous fluid called cytosol. The cytoskeleton is a matrix of proteins that helps give the cytoplasm its structure and holds the organelles in place. So organelles. This means little organs, and they are very organized and highly specialized structures that carry out specific functions that keep the cell alive. Some have membranes and some do not. So just like the cell's very own plasma membrane, organelles with membranes also keep the fluid like cytosol and any neighboring organelle bits from entering its domain. The reason for this is that these organelles have their own chemical activity going on that is not compatible with each other. It has to remain separate in order to function properly. The main membranous organelles we find in cells are the endoplasmic reticulum, Golgi complex, mitochondria, lysosomes, and peroxisomes. So a great example of why these are self-contained little organs is that lysosomes make protein-destroying enzymes, and they digest proteins that are no longer needed. So you can imagine what would happen to all the needed proteins in the cell, like the cytoskeleton and transporter proteins in the cell's plasma membrane, if these enzymes weren't contained within the lysosome's membrane. In fact, we know that leaky lysosomes can kick off pathways responsible for premature cell death. So membranous organelles have membranes for very, very good reason. The organelles without membranes in the cytoplasm are ribosomes, centrioles, vaults, and proteasomes. And just like the membranous organelles, they have specific jobs that keep the cell functioning. So let's start with the membranous organelles. The endoplasmic reticulum, or ER, is the largest of the organelles. It has rough and smooth components. The rough ER kind of looks like folded membranous ribbons, and it's studded with ribosomes, and that's what kind of makes it look rough. It's connected to the nucleus's outer envelope and has an important role in synthesizing and packaging proteins that will either be used by the cell or end up exiting the plasma membrane and into the extracellular environment. We'll get into more detail about this process when we get to ribosomes. But for context, cells that need to produce a lot of proteins, like liver cells, will have larger, more robust, rough ER. The smooth ER does not have ribosomes attached to it and looks more like smooth tubes. It packages the newly synthesized proteins for transport. It also metabolizes lipids that you find in membranes and steroid hormones and also prepares them for transport. And when I say transport, I mean they kind of form bubbles around the proteins and lipids that look like escape pods, which are then pinched off into the cell universe. I mentioned them earlier when talking about proteins in the plasma membrane. These pods are called transport vesicles, but before the proteins and lipids packaged inside of them can go about their business, the transport vesicles carry them to the Golgi complex for further refinement. The Golgi complex is also known as the Golgi apparatus, also Golgi body and sometimes just Golgi. It's named after the pathologist who discovered it, Camillo Golgi. He made a few contributions to neurobiology, so we'll circle back to him again in the next episode when we look at neurons. So what is the Golgi complex? Well, it resembles about 4 to 8 stacked pouches that look like they've had the wind knocked out of them. Or, in the vicinity of my world, what 4 to 8 soccer balls look like after French Mastiffs get a hold of them. These flat pouches are called cisternae, and they play an important role in packaging, sorting, and transporting proteins and lipids to many other destinations of the cell, as well as into the extracellular space where they can enter the bloodstream. So, remember our transport vesicles, or escape pods as I like to think of them? They're full of newly synthesized proteins and lipids and have made their way to the Golgi complex from the smooth ER and fuse with its membrane so it can release the molecules it contains into a Golgi pouch. The protein and lipid molecules make their way through the pouches, and as they do, they are refined into their final form. Then the Golgi separates them and sorts them and repackages them based on their destination. It then produces more escape pods to send them off. It's a rather elaborate system because each protein and lipid is designed and synthesized for a specific function, and the Golgi has to know what that is so it can send it off to the right place. The Golgi complex is pretty special. It plays a big part in the homeostasis of the cell, and its dysfunction is implicated in neurodegenerative, cardiovascular, and infectious diseases, as well as cancer. So we very much need these tiny deflated soccer balls to work properly. Okay, so moving on, we have the mitochondria, and it is known as the cell's powerhouse. It draws energy from the nutrients in our digestive food and converts it into energy our cells can use. This energy is called adenosine triphosphate, or ATP. ATP is the fuel cells need to do their business, and by extension, what our whole bodies need to do everything. ATP is generated when glucose is broken down in 3 chemical reaction stages, called cellular respiration. But it isn't synthesized for use until the 3rd stage, where the mitochondria converts ATP into fuel through proton and electron wizardry called oxidative phosphorylation. An excellent example of mitochondria being its powerhouse self is how it provides the horsepower needed for a sperm cell to not only make its epic journey to a fallopian tube to fertilize an egg cell, but also has to outrun hundreds of millions of other sperm cells in order to be the winner. So thank your mitochondria because you are the sperm that won. Other than ATP, the mitochondria's next claim to fame is that it holds the cell's self-destruct button. It's a process called apoptosis, and it occurs when a very useful chemical inside the mitochondria is purposely leaked out into the cytoplasm. It triggers a biochemical reaction that shreds the cell into rubbish, and that will get cleaned up by other types of cells. Every cell that has a mitochondria has this mechanism. When a cell is at the end of its life, because it's no longer useful or there's something wrong with it, like it's under attack by a virus, infection, or anything that could threaten homeostasis. The mitochondria kicks its own bucket. This type of cell death, apoptosis, is actually a very normal and necessary part of cell turnover. We'll visit this mechanism again when we get to nervous system cells called astrocytes and microglia. As a side note, there is another type of cell death called necrosis, and that is due to irreversible injury to the cell, like disease, stroke, or other physical trauma that triggers structural changes over many hours and prevents the cell from functioning. There are some caveats as to when this type of cell death is considered necrotic, and if you'd like to know more about the difference between these two types of cell death, I highly recommend watching Dr. Najib's lectures here on YouTube. I'll include a link in the show notes to his lecture on necrosis, in which he explains both necrosis and apoptosis. Okay, back to mitochondria. Something else that is noteworthy is that mitochondria contain their own DNA distinct from the cell's genome located in the nucleus. This may be left over from an ancient version of itself as a single-celled prokaryote. It appears to only encode proteins that are necessary in the oxidative phosphorylation process, but it's kind of wild that our mitochondrial genomes may have evolved from some ancient prokaryotic bacteria that is yet to be identified. On a slightly more distressing note, mitochondrial DNA mutations are the source of many, many disorders involving most of the body systems, if not all. The next membranous organelle we'll look at are lysosomes, and as I've alluded to already, lysosomes are the cell's digestive system. They digest unwanted bits both outside and inside of the cell by way of a few different processes. To clean up unwanted extracellular macromolecules, such as pathogens, lysosomes will make enzymes that break them down in a process called endocytosis. To clean up large extracellular bits like dead cells and invaders like bacteria, lysosomes that live inside specialized cleanup cells called macrophages will digest these bits in a process called phagocytosis. Inside the cell, lysosomes will also digest worn out or damaged organelles in a process called autophagocytosis. But interestingly, lysosomes won't completely digest an entire organelle. It will leave behind components that can be reused by the cell, which leads to an important side note, and that is that organelles are renewable. So don't worry that they become snacks when they cease to function, as they should. In most cases, it does the cell more harm to not break them down. Structurally, lysosomes are small and round, but vary in size depending on what they're digesting, and there are roughly 300 of them going about their business in any one cell at any one time. When lysosomes aren't functioning properly, it can lead to disease states. Other than leaky lysosomes mentioned earlier, it's also possible for genetic mutations to inhibit the production of certain lysosomal enzymes, which causes the lysosome to accumulate molecules that would normally be digested. This overaccumulation leads to enlarged lysosomes that disrupt the normal functioning of the cell, and this can be extremely problematic. This has a name. It's called lysosomal storage disease, and more than 50 variations have been identified. One example is Tay-Sachs disease, and this is when there is an overaccumulation of molecules in lysosomes in nerve cells, and this leads to nervous system degeneration. So like the other organelles, we need these tiny cannibals to work properly. And the last of the membranous organelles are peroxisomes. They are even smaller than lysosomes, but about as numerous, and they also contain a number of enzymes that break down molecules for all kinds of purposes. The main difference, though, is that lysosomal enzymes are hydrolytic, which means acidic, and this acidity is what breaks down the materials that are no longer useful. But enzymes in peroxisomes are oxidative, and these oxidative enzymes, though they break down substrates, sometimes they do so to turn them into something useful instead of getting rid of them or recycling them. For example, fatty acids are broken down by peroxisomal enzymes and turned into metabolic energy. Another one of their functions is a kind of detoxifying superpower. They do this by creating oxidation reactions with potentially toxic molecules. and then by chemical magic strip the molecules of their toxicity. But get this, in the process of doing this, peroxisomes end up producing hydrogen peroxide, which is also toxic. So they have to release an enzyme that converts the hydrogen peroxide into water and oxygen before the hydrogen peroxide escapes into the cytosol and destroys the cell universe. I feel like there could have been a less stressful design here because that is some serious wax on, wax off duality. I'm a visual person, so every time I see someone drinking alcohol, I imagine the peroxisomes in their liver cells turning into hundreds of tiny Mr. Miyagi's sidekicking the toxic alcohol compounds into oblivion, but then have to smack down the ghost vapor they leave behind into water and oxygen. Just to be clear, that is not permission to drink alcohol excessively. There is a lot more going on in the liver to break down alcohol, and overconsumption can inhibit the liver's ability to produce all the enzymes needed to process it, as well as inhibit other organelle functioning, including mitochondria, which incidentally are also needed to break down alcohol toxicity in the liver, so it would be very helpful to your liver if they remain functional. Okay, thank you. Okay, next we'll look at the nucleus and then get to the non-membranous organelles afterward. Most of them will make more sense after we discuss the nucleus anyway. Okay, so the nucleus is the largest membranous beast inside the cell universe, but it's not an organelle. It's its own entity, usually located somewhere near the middle of the cell. It is where your deoxyribonucleic acid lives. You'll know it as DNA, and it is all of your genetic information, and it's neatly packaged into chromosomes with other nucleic proteins. Each nucleus in most human bodies has 23 pairs of chromosomes. That's 22 pairs of autosomes and 1 pair of sex chromosomes. There are some instances when humans can have chromosomal anomalies. These anomalies are organized by either numerical anomalies or structural anomalies. Structural chromosomal anomalies include deletions or duplications to parts of the chromosome, and these affect the amount of genetic material in the cell. Numerical chromosomal anomalies are when someone has an extra chromosome or is missing a chromosome. This is called an aneuploidy. A common aneuploidy is a trisomy, and a trisomy is having a third copy of a particular chromosome. Down's syndrome is one example of a trisomy to chromosome 21. So people with Down syndrome have a third copy of chromosome 21 in each cell instead of just the pair. Klinefelter syndrome is another example of a trisomy, but this affects the sex chromosomes. Klinefelter syndrome is when someone born with an XY chromosomal pair has an extra X chromosome, so their chromosome makeup would read XXY. It is also possible for XY humans to have an extra Y chromosome, and this is called 47XYY syndrome or Jacobs syndrome. Another example of an aneuploidy is a monosomy, and that is the loss of a chromosome in each cell. An example of a monosomy is a Turner syndrome. People who are typically born with XX chromosomes but have Turner syndrome will only have one copy of the X chromosome in each cell instead of the typical XX chromosomal pair. There are many types of aneuploidies, but only a few of them are compatible with life, and those that are will have differing impacts on developmental and/or reproductive outcomes depending on the genetic features of the chromosome affected. As I mentioned already, the genetic material inside the DNA is the blueprint your cells use during replication. It contains the information each cell needs to become the cell it's supposed to be. DNA also guides protein synthesis, including enzymatic proteins, and both proteins and enzymes are crucial for the structure and chemical activity of the cell. So in a somewhat indirect way, the nucleus, by way of DNA, is responsible for the cell's infrastructure and its economy. So you could think of the nucleus as a kind of mayor of the cell universe. We'll take a closer look at protein synthesis in a minute when we get to non-membranous organelles called ribosomes. But I just want to briefly mention two other important terms associated with DNA that you'll often hear bandied about in health and wellness circles. The first is genome. The human genome is the complete set of genetic material encoded as DNA, which is duplicated and housed inside of our chromosomes. Something worth looking up is the Human Genome Project. This colossal, internationally collaborative project sequenced, I think, about 90% of all human DNA, and it took about 13 years to accomplish this. It's now an incredibly extensive resource for human biological research in the realms of health and disease. It's worth reading about. The second term is epigenetics. Epigenetics refers to a gene modification due to environmental influence. That could be external environment or internal environment, and loosely what happens is this environmental influence causes chemical tags to be added to a DNA sequence or to its related proteins. It won't change the DNA sequence itself, but it will change how your body reads the DNA sequence. For example, depending on your genetic predisposition, you could change the way a gene is expressed by overexposure to stress, or even by one catastrophic life event. This could result in a gene behaving differently that could in turn increase your risk for clinical levels of depression or anxiety, or a myriad of other health issues. This isn't going to be the same for every person, but every person is subject to epigenetic influence in some way. And from what I understand from the literature is that this is reversible in many instances. Lifestyle changes or reducing or extricating yourself from toxic environments can undo epigenetic influence, and the gene will return to behaving as it should. A former middle school chum of mine is very gifted at communicating the complexities of genetics in digestible sound bites and has co-authored a book on a gene editing process called CRISPR. It's very much an intro-level read, so if you're interested in learning more about how genes operate, I recommend giving it a go. I hope to collaborate with her on an episode devoted to genetics, epigenetics, and gene editing, but maybe let me know in the comments if this is of interest to you. If so, I'll try to get that organized very soon. Regardless, I'll post a link to her book in the show notes. Okay, on to non-membranous organelles, or organelles without membranes. And to recap, these are ribosomes, centrioles, vaults, and proteasomes. I'll try to wrap these up quickly-ish. First up is ribosomes. If you remember, I said earlier that rough endoplasmic reticulum is studded with ribosomes, and the rough ER surrounds the nucleus, which is handy because ribosomes play an important role in protein synthesis, and protein synthesis begins in the nucleus with DNA. Through a process called transcription, a particular code for the protein meant for synthesis is transcribed into messenger ribonucleic acid, or mRNA. You will likely recognize the term mRNA as it is at the center of recent advances in vaccinology. So mRNA exits the nucleus and is delivered to ribosomes, and with spy-like precision, the ribosome cracks the protein code and translates it into amino acids, and with the addition of other RNAs, enzymes, and energy, ribosomes link chains of amino acids into sequences dictated by the DNA instructions until the protein is born. Then the rough ER releases the protein into its own gel-like substance that's like cytosol but contained within its own membrane. There, it's folded into its almost final form and sent to the smooth ER for packaging and transport to the Golgi complex, as explained earlier. The point of protein synthesis is to make proteins that will become something useful to the intra or extracellular function. Maybe it's going to become part of a membrane. Maybe it's going to be a hormone. Just know that ribosomes take care of the bulk of protein synthesis. Okay, on to centrioles, and by extension, microtubules. Centrioles are non-membranous organelles made up of microtubules, structured in a kind of ringlet formation. They are located at the center of the cell, also called the centrosome, near the nucleus. There are usually two centrioles perpendicular to each other, and this pair play a role in microtubule formation. Something I find interesting about centrioles is their shape. The arrangement of microtubules in a centriole is something quite beautiful and symmetrical. If you slice one in half and looking at its cross-section, there's just something about this shape that is familiar. It's called a cartwheel, but it almost looks floral or star-like or galactic, which, I don't know. I'm always a bit in awe of the way patterns are repeated in nature, and it's always surprising to me that it's found even in something so microscopic. In this instance, the microtubules are gathered in triplet and arranged in a ring. I'm not quite sure the benefit of this particular pattern to the overall function of the centriole, but a study by Wang and colleagues found that this pattern, specifically the arrangement of microtubules in triplet, seemed to promote centriole stability and longevity, so they were able to carry out their functions properly, mainly to form and organize microtubules. Okay, you might be shouting at your screen right now, "This is too much detail," and I apologize, but I promise there's a point to understanding this. For one, it's so you can understand that even the tiniest, seemingly benign parts of your cells are in fact integral to homeostasis and the health and well-being of your body. And secondly, when these parts aren't working properly, it can lead to disease states. Defective centrioles, in particular, are implicated in cancers and genetic conditions that lead to diabetes, obesity, heart disease, and a host of other disease states. If centrioles can't form and organize microtubules properly, that sets off what scientists love to call a cascade of events, and I've mentioned this earlier. A cascade of events is like a domino effect. If centrioles aren't working, microtubules won't be able to function properly or at all, and they have a ton of important functions in cells. And if those important functions don't happen, then tissue, organs, and whole body systems become compromised. At this juncture, it might be good to know what microtubules are and what they do. Microtubules are not organelles. They are long tracts that radiate out from the centrosome, or cell center, toward the plasma membrane, and they have all sorts of functions. One of their functions is involved in mitosis, which is related to cell division. Another is the formation and anchoring of the flagella and cilia, which are tiny hair-like structures involved in cellular movement. Another is positioning organelles in the cytoskeleton and maintaining the shape of elongated cells like nerve cells. Another really fascinating function of microtubules in nerve cells is the transport of material inside the long skinny fiber called the axon that protrudes from the cell body. We'll learn more about that in the next episode. It's kind of adorable and slightly unexpected. You're not going to want to miss that, so stay tuned. Anyway, centrioles have important functions, and they're mostly related to the formation and organization of the multifunctional microtubules. We will be revisiting these beauties throughout the series. Okay, next up are vaults. Vaults are sort of enclosed barrel-shaped protein shells, and they are very, very tiny. Each of our cells contain roughly 100,000 of them. We're not certain yet what their purpose is, but their shape and size is similar to the pores in the nucleus membrane. They're also hollow inside, so they could be involved in transport of some kind. It's possible, but not yet confirmed, that they carry mRNA from the nucleus to ribosomes for protein synthesis. For now, though, they're still a bit of a mystery. The last non-membranous organelle we'll discuss is proteasomes. These are cylindrical-shaped complexes that love to devour defective proteins. Sometimes the cell doesn't get everything right, and during protein synthesis, proteins can become misfolded. When they are, the rough ER tags them with something called ubiquitin, which signals to the cell that this protein is defective. These defective proteins are then sent on to the many proteasomes floating in the cytosol. Proteasomes recognize the ubiquitin tag, and its job is to unfold the protein, release enzymes to degrade it back down into amino acids, and the amino acids are then reused for protein synthesis or to create energy. The ubiquitin is also released and reused. I mean, this is ISO bazillion level quality assurance. It's like we're packed with trillions of tiny environmentalists. Our cells are so efficient and so responsible and so economical and work so hard to take care of each of their little universes. And as they do, they take care of us. So that's it for this episode. It was a lot today. Thanks so much for sticking with me to the end. But hopefully you're starting to get a better idea of just how much work your cells are doing to keep your body in balance, to keep it in homeostasis, and doing so with tremendous complexity and efficiency. I hope your cells have inspired you to take a little extra care of you, your surroundings, and any other chunks of cells that share your living space with you. I'll leave you today with one of the sweetest cell love stories I've ever read. It comes from Dr. Jill Bolte Taylor. If you're unfamiliar with Dr. Taylor, she is a neuroscientist who's suffered a massive stroke in her left hemisphere. Her book, My Stroke of Insight, is her incredible story of recovery, and in it she describes her cells as trillions of geniuses and makes a point to thank them every morning and night for their hard work. In her words, "I care enough to say it out loud. Thank you, girls. Thank you for another great day. And I say it with an intense feeling of gratitude in my heart." End quotes. Thank you, Dr. Bolte. That is solid advice. Your homework for this week is to thank your cells for doing their best to take care of you. Also, if you like, take a look at the origin and evolution of cells. There are a few theories, but I'll drop one link in the show notes and you can go from there. There's also a really wonderful paper by Brandon Ho and colleagues, and they have calculated the number of protein molecules in the average cell. It's a lot. Bravo, Brandon and team. I'll put a link to that in the show notes below. Also of note, and very exciting, as I'm writing this episode, Dr. Carolyn Bertozzi and two of her colleagues have just won a Nobel Prize in Chemistry for her prolific work developing chemical pairs that bond no matter what chemical reactions are happening around them, and that has created a way for researchers to study how molecules interact with living cells without interfering with natural biological processes. This seemed appropriate to mention in this episode because this chemistry has made it possible to study living animal cells. A hearty congratulations to Dr. Bertozzi. By the time this gets published, this will be very old news, but it's still very exciting. At the end of the show notes or pinned in the comments, you'll find a list of papers and books I sourced to write this episode, so you may find something of interest there as well. Before we end today, just a gentle reminder to do what you can to support STEM education. There is a list of organizations in the show notes you can connect with, and do let us know in the comments of any other projects or organizations in your communities that support STEM education and research resources that aren't mentioned in the list so others can contribute as they are able. If you liked this episode, please click the like and subscribe buttons and the notification bell if you haven't already. Please share it with your friends, neighbors, and family, and anyone you think would benefit from understanding their body a little better. Until next time, take care out there. Sorry, I have a bit of a cold. I can't breathe through this. Okay, so let's start with the membranous organelles. That's going to be hard to say with a stuffy nose. Membranous. So sorry. The membranous organelles.